Welcome geniuses, I'm Genie, your best buddy for A-levels. In this channel, we'll bring you to explore the secret formula behind success.
For this video, I'm going to cover AS topic 10, infectious disease, and I'm going to focus on learning outcome 10.2a about how penicillin act on bacteria. So what is the mechanism of antibiotic? Number one, you have to understand that antibiotic act on bacteria and fungi. And number two, the action of antibiotic can be bactericidal. So it sounds like suicidal, is to kill, or bacteriostatic. So the static basically means make them immobile, not moving or not multiplying, therefore buying time for your immune system to recognize and attack. So by looking at the diagram, can you identify the methods or the actions of antibiotic? Number one, to kill a bacteria or to stop a bacteria from multiplying, we can inhibit the protein synthesis by targeting the ribosome of the bacteria. So there will be no translation, no protein synthesis, so the bacteria will stop multiplying and die. Number two, we can inhibit the nucleic acid synthesis. So when there's no DNA replication, there will be no binary fission, then there will be no reproduction for bacteria. Number three, we can see that antibiotic can inhibit the cell wall synthesis. So when there's no new cell wall, the cell wall becomes weakened, and the bacteria will actually burst when they cannot withstand the osmotic pressure. And number four, we can see that the membrane function gets disrupted, so the content of the bacteria will start to leak out. And the last example will be antibiotic can actually act as an inhibitor to block the pathway and inhibit metabolism of the bacteria, therefore causing the bacteria not being able to survive. So by looking at mechanism 1 to 5, some of them will belong to bactericidal and some of the action belongs to bacteriostatic. So which action does the antibiotic penicillin applies? You will see how penicillin actually kills the bacteria by weakening the cell wall. If you're going to start the description of how penicillin acts on bacterial cell, we will start with the introduction that penicillin acts on growing cell. So it means that antibiotic penicillin only acts on actively growing bacterial cell and not the dormant ones. So when the bacteria is growing, the bacterial cells needs to elongate, but with the rigid cell wall, which is the peptidoglycan cell wall, the bacterial cell is unable to expand. So what happens here is, and we know that the peptidoglycan is actually made up of lots of cross-links to hold the structure. During growth, the bacteria will secrete enzyme autolysin that will actually break down the cross-link, making holes on the peptidoglycan cell wall. This allows the cell wall to be loosened up and can expand easily when the bacterial cell grow during the uptake of water. So point number 2, autolysin is secreted to punch hole on the peptidoglycan cell wall. So point number 3, how does the antibiotic act on the bacterial cell, thus killing them? Antibiotic penicillin act as a inhibitor on enzyme glycoprotein peptidase. And what is so important about this enzyme? This enzyme is responsible to build a crosslink within the broken peptidoglycan cell wall. Without this enzyme, the crosslink within the peptidoglycan cell wall is not repaired or is not formed. Therefore, the cell wall becomes weakened. We don't say that the bacteria do not have a cell wall, but instead, the cell wall is weakened. Then what happens if the cell wall is weakened? As the bacterial cell is constantly in a watery environment, so water will be coming into the bacterial cell by osmosis. So usually the bacteria will just expand and they will just grow and patch up the cell wall. But in this situation, the cell wall is no longer able to withstand the osmotic pressure, or sometimes it's known as pressure potential. Therefore, it will burst, or sometimes it's known as lysis. So this is how antibiotic penicillin causes the death of a bacteria. So let's look at sample past year question. So for this question, the question stated that antibiotic penicillin prevents the formation of crosslink between the peptidoglycans. So in this case, then they mentioned the enzyme transpeptidase, which is sometimes also known as the glycoprotein peptidase. So how would you describe the action of penicillin? So we know that it's the enzyme inhibitor. So the enzyme can no longer form the crosslink and therefore weakening the bacterial cell wall, which is correct. And it will work at any stage. So the any stage includes the active and the non-active part of the life cycle. So this is not really qualified. So the answer is only 1 and 2. This question requires not only your understanding about the action of antibiotic, it also requires your understanding about the structures of bacterial cells. So when we are talking about the actions of antibiotic, remember I said that antibiotic can either inhibit protein synthesis, can inhibit DNA replication, or inhibits the cell membrane function, or weakening the cell wall. So now the question is asking, which of these antibiotics will affect only the bacterial cells? That means when you are trying to identify the correct answer, you have to find out the characteristics or the structure that's exclusively found only in bacteria. Therefore, it will only inhibits the bacteria replication or the bacteria's metabolism without affecting the eukaryotic cell. So inhibits RNA polymerase. This one is meant for the process of transcription to produce mRNA. This is found in both prokaryotic and eukaryotic cell, so it's wrong. streptomycin inhibits 70S ribosome. So some of you will be thinking that, true, 70S ribosome is found in bacteria, but the thing here is, it's also found in mitochondria and chloroplast. So this is a trick question. If you can think of this, you might have doubt about this question and you will eliminate. Point number 2. Neomycin inhibits DNA synthesis. Yes, this one in both prokaryote and eukaryote. And the last one, ampicillin, inhibits peptidoglycan synthesis. This one is a cell wall, and cell wall that is found only in prokaryote. Therefore, the answer is definitely D, 4 only. And the last question here shows that antibiotic inhibits the formation of cross-link between the molecules that form cell wall in bacteria. So take note, molecules within the cell wall. What comes into your mind? So the first statement says that the bacterial cell is destroyed by osmotic lysis. True. Now, question 2 sounds like it's correct because it's a cell wall. Cellulose molecule forms a cell wall, and if they cannot form hydrogen bonds, then they cannot withstand the pressure. But take note, cell wall in bacteria is not made up of cellulose, but instead is made up of peptidoglycan. So statement 2 is definitely wrong. And the cell wall is no longer selectively permeable. Now, cell wall is never selectively permeable. Cell wall has been always fully permeable. Therefore, statement 2 and 3 is wrong, and the answer is only 1. So it would be C. For more understanding on why antibiotic only works on bacteria but not on virus, and how bacteria gains antibiotic resistance, do contact the following number for more information on group and private tuition. Thanks for watching. Thank you.
That's all for today's video. If you're interested in more genuine sharing by other geniuses, please subscribe to our channel and don't forget to turn on the notification bell, ding dong. Also, if you're struggling with 1 or 2 past year questions and the mark scheme just doesn't seem to help, Genie has got you covered. Feel free to let us know what question it is by filling in the Google form linked in the description below. GenieUs Hub will get genius teachers to fulfill your request for the solution. Genie will see you next time. Bye-bye.